audio is dominated by the continuous soft hum of HEPA air filtration systems, creating a sterile.
The markers align with the Archosaurian profile.
Confirmed. Proceeding to sequence the next batch.
Process A is clear for integration.
Confirmed. The parameters are well within the acceptable range for the reptilian samples.
Flow rates are stable. We have achieved 99% purity on the reptilian samples.
As you can see, the B pathway leads directly to the long-term storage tanks.
Yes, optimizing the integration timeline.
Dispatch confirming manifest for Process A. Routing to Tokyo and Berlin research facilities. Containers secured.
This cluster here suggests a strong correlation with the target sequence.
The integrity of the cryopreserved material is remarkable.
The proliferation rate in chamber 3 is exceeding projections.
The biobank samples are proving incredibly viable.
The genomic sequencing confirms a perfect match with the biobank samples.
And the population models show viability for release by Q4.
Post-thaw viability staining confirms the successful recovery of the samples. The bright green fluorescence
These vials represent future stock. They are the key to unlocking critical medical breakthroughs.
Quality check on CryoRack 4 is complete. No anomalies detected.
Acknowledged. Releasing robotic arm for return.
Extraction of reptilian genetic material underway.
Lock sequence engaged.
Confirmed. All tags reading green. Seal the container.
As you can see, the green markers indicate high viability for batch gamma 7.
What about the samples moved yesterday from Sector 4?
Backup engaged. Check synchronization across the board.
All systems reporting nominal. Freezers holding steady.
The data streams from the South American Biobank are stabilizing.
Cross-reference that with the Process A viability metrics immediately.
The welds look clean on this section.
Agreed. The automation is holding the parameters perfectly.
We are verifying every micrometer of the cut to ensure the structural integrity required for ultra-low temperature environments.
Pressure levels are dropping according to spec.
We're almost at target vacuum on tank 4.
System check complete. Everything is nominal.
Test sequence complete. All parameters nominal.
Data confirms structural integrity. We can approve the batch.
The high-sensitivity scan confirms the seal's integrity, crucial for protecting the invaluable genetic material stored within the cryotank.
By scanning the QR code, the technician verifies the contents, linking the kit to the tank's digital service record, ensuring long-term operational readiness.
Move row 3 to staging bay 7.
Acknowledged. Dispatching movers now.
Hoist engaging on tank 4.
Copy that. Alignment looks good. Proceed with securing.
The vibration data confirms the casing's integrity. The resonant frequencies are nominal, ensuring the biobank materials are secure for transit.
With the sensors synced, we can monitor external conditions in real time, ensuring the cryogenic environment remains stable throughout transit.
Control, Module 42 is approaching the loading ramp. We are clear for Transport Sequence Alpha.
Please now bring it down 5 centimeters. Secure the forward clamps.
And with this new tank fully online, our integrated monitoring confirms optimal status across all parameters.
The data stream from the reptilian samples confirms the viability rates.
And the global impact markers are responding in real time.
This facility represents our commitment to the future.
The biodiversity preserved here is irreplaceable.
Look at all the different reptiles they are saving.
It says this tank keeps them frozen forever.
It's incredible technology, isn't it?
A vast, invaluable genetic library. A technologically advanced sanctuary